Today I'll be discussing folliculogenesis. Folliculogenesis is the process of ovarian follicle development from primordial follicle to a mature follicle capable of ovulation. So I'll be discussing this topic under headings of embryology, the hormonal interplay, and then the clinical aspects of it. So, during fetal life, this embryology part is very important when it comes to the folliculogenesis. As you know, the process will start during the fetal life. The human ovary during fetal life passes through 4 stages. The first one is the indifferent stage of the gonad, where the gonad has not differentiated. Then the differentiation occurs. Then the stage of ogonial multiplication and oocyte formation, and then follicle formation. The indifferent stage of the gonad, initially at 3 weeks of gestation, there is appearance of germ cells, and then 4 to 5 weeks there will be migration of germ cells. 5 weeks there is appearance of genital ridge. Then 6 weeks, till 6 weeks it is indifferent gonad. So 6 weeks there will be completion of indifferent stage. Then 6 to 9 weeks there will be differentiation of the gonads to ovary or testis. Then the differentiation of the gonad. This differentiation to testis or ovary will occur according to the genetic constitution of the fetus. And if it is a male fetus, then the sex determining region on the Y chromosome and SRY box 9 gene will lead to Sertoli cells, development of Sertoli cells. And when there is no Y chromosome, then the W and T4 and R spondin 1 gene will lead to development of granulosa cells of the ovary. Then the stage of ogonial multiplication and oocyte formation. These germ cells will develop, multiply by mitosis, and then reach 6 to 7 millions in number at 16 to 20 weeks of gestation. So this is the maximum number, and after which the number will start declining. They will undergo attrition and they will start declining in number. This is the maximum number which is seen at 16 to 20 weeks of gestation. Then the germ cells, they give rise to
Recording in
oogonia
progress.
by 7th week. The oogonia form oocytes at first meiotic division, which gets arrested in the Then the follicle formation. The follicle formation will start at 18 to 20 weeks. The primordial follicle consists of oocyte, which is arrested in prophase of meiotic division, which is covered by a layer of spindle-shaped granulosa cells. So this is the, as you can see in this picture, the germ cells will arrive at the gonad, then they form a syncytium, then they will be invaded by the granulosa somatic cells. Then the encapsulation, this is the oocyte, and the encapsulation of the oocyte by pregranulosa cells. And when they are encapsulated, they are called the primordial follicles. That is the oocyte covered by the granulosa cells. So this primordial to pre-ovulate, once the primordial follicles develop, then the development continues to primary, secondary, and to pre-ovulatory follicle. It will take up to 1 year time. Then from primary to pre-ovulatory follicle may take up to 85 days. So this is the number of germ cells in relation to the age. So initially there will be about 100 of germ cells. They will multiply by mitosis and reach the maximum number by about 20 weeks of gestation, that is 6 to 8 millions. And then the number will come down. At birth, the baby will have 1 to 2 millions. And then the atresia will continue. And at puberty, we are left with only about 3 lakhs. And then with every month, the atresia degeneration will continue, and then in the whole reproductive age, a woman may be able to ovulate 400 follicles. So for every one follicle to reach the stage of ovulation, the other 1,000 follicles will undergo atresia development and then atresia at certain stage. So in detail, the gametogenic follicles, they have, each one have a oocyte and it is a site of oogenesis. This primordial follicle is by 7th month of embryonic development within the cortical stroma. It is made up of first order oocyte surrounded by follicular cells, these cells being covered by basal membrane. Then comes the primary follicle, wherein the surrounding follicular cells are transformed to cubic cells. Then the secondary follicle, that is a single follicular layer, multiplies, and then their differentiated epithelial cells form stromal fibroblasts and is associated with capillaries. These epithelioid cells together form interna theca of the follicle. So a secondary follicle that possesses the internal theca is called a preantral follicle. Then the secondary follicle to tertiary follicle develop. This is a antral follicle wherein there is a cavity or antrum and then the theca externa and then a theca interna. So this is the same as we have discussed. So as you can see, a ovarian follicle structure, it will have a oocyte surrounded by a layer of cells. A primordial follicle will contain the oocyte with a single layer, whereas as a follicle develops, then it will develop a zona pellucida, a granulosa, and then theca interna and theca externa layers. This is a Graafian follicle structure where the ovary follicle is ready to rupture, that is just before ovulation. Then it consists of a secondary oocyte that is formed after first meiosis is completed. It is surrounded by zona pellucida, then this cumulus oophorus and a fluid-filled antrum and theca interna, which is vascular, and theca externa, which is a connective tissue capsule. So this picture, we can see the follicle development from primordial follicle to the Graafian follicle and the ovulation, and then the meiotic events which occur in the as the follicle progresses from oogonium to the by mitosis. They'll give rise to oocytes, primary oocytes. Then they are arrested in the prophase 1. Then till before ovulation, they are arrested in the prophase 1. And just before ovulation, the meiosis is completed and the polar body is released. And then after ovulation, after the sperm enters the oocyte, the meiosis 2 is completed. And the second polar body is released. In summary, the follicle is the functional unit of the ovary. One female gamete, that is the oocyte, is contained in each follicle. The granulosa cells produce hormones, estrogen and inhibin, that provide status signals to the pituitary and brain about the follicle development. So coming to the hormones, in all these development of the follicle from primordial to the ovulation, all these 5 hormones play important role. They give a positive and negative feedbacks to regulate the folliculogenesis. They are GnRH by the hypothalamus, FSH and LH, and estrogen and progesterone. So in the early stages, wherein primordial to very pre-antral, early antral, the development is not gonadotrophin dependent, it is independent. Whereas once the follicle reaches the antral follicle stage, the growth is dependent on the gonadotrophins. So a follicle, to reach the stage of ovulation, it has to undergo recruitment, selection, dominance, and then ovulation. So for a single follicle, as you know, most of the follicles, the fate is atresia, that is a degeneration. Most of the follicle will undergo degeneration. Only very few follicles will reach the stage of ovulation, all through recruitment, selection, dominance, and ovulation. So the recruitment, uh, there are three theories proposed for follicular recruitment: continuous, single recruitment, or a follicular wave theories. So continuous recruitment, it is it occurs throughout the menstrual cycle and is independent of gonadotrophins. So the follicle destined to ovulate is said to be selected by chance from continuous supply of antral follicles by virtue of being at the right stage of maturity to respond to the rise in FSH and subsequently LH that occurs following luteal regression. Then the second theory is a cyclic recruitment, which states there is a recruitment of a cohort of follicles in the late luteal phase or early phase of each cycle, early follicular phase of each cycle. So an ovulatory follicle is selected from a single follicular cohort that emerges following luteal regression. The antral follicle, why only one follicle is selected among that cohort? That depends on the concentration of the estradiol and the androgens in that follicle. So the next theory is the follicular wave theory. So in contrast to the notions of continuous recruitment or a single episode of cyclic recruitment, there are cohorts of antral follicles referred to as waves. So they emerge multiple times during the menstrual cycle. And recently, the studies using abdominals and transvaginal ultrasounds, they have supported the notion that multiple waves of antral follicles develop through the menstrual cycle in a healthy woman. So this picture shows this above picture in between two ovulations. This is interovulatory. This is one ovulation and this is the other ovulation. So in between two ovulations, there are two follicular waves. So this is the one wave that is growing and the ovulation is happening, and this is one more ovulation, and in between there are multiple waves of follicles growing. But they are not reaching the stage of ovulation. And here in the picture down, there are, in between the ovulation, there are three waves. So some are major waves, we say, some are minor waves, wherein a dominant follicle is developed, that is called a major wave, and whereas some waves which may not reach to the stage of dominant follicle, we call it as a minor waves. This is a follicular wave theory. So this follicular wave theory that forms the basis for all the random start ovarian stimulations, because wherein we do the stimulation at any point of time in a menstrual cycle, we do that depending on this theory of continuous follicular waves. So in this picture, this is a natural cycle wherein the FSH concentration, after it is above the threshold, and in this window there is a follicular recruitment and selection and dominance happening. But that means once the FSH concentration is above this threshold, the follicle gets selected and follicle gets dominance, and then ovulation happen. And below that concentration, below the threshold, there won't be any follicular development, and all these will undergo atresia. So this is a two-cell two gonadotrophin theory, which is very important and forms the basis of our folliculogenesis, wherein the LH acts on the LH receptors of the theca cell of the follicle. Then through cyclic AMP and protein kinase A, the cholesterol is converted into androstenedione. This androstenedione in the granulosa cell under the action of FSH and the FSH activating the P450 aromatase, the androstenedione is converted to estrone and then to the estradiol. So FSH is crucial for inducing the aromatase enzyme, which is responsible for converting androgen to estradiol. In later stages of follicular growth, the estradiol and activins enhance the action of FSH. Then the role of LH. The LH, as you have seen, it will be in the theca cells. The progesterone is androstenedione is synthesized and which is converted to aromatase in the granulosa cells, and then and under aromatase the androstenedione is converted to estrogen in the granulosa cells. So LH also has a threshold and a ceiling effect. So below this threshold of LH, there won't be any follicular development, then inadequate thecal androgen synthesis, and hence reduced granulosa aromatization to estrogen. So there won't be any full oocyte maturation. Whereas when there is a ceiling that is above this level, there will be LH receptor downregulation and suppression of granulosa cell proliferation, then follicular atresia. Only in this optimal window, there will be optimal follicular growth and development and full oocyte maturation. So this is all the timeline which we have discussed. There is a resting follicles which stay in that phase for more than 120 days, and once they are activated, they'll reach the preantral follicle. From preantral follicle to the ovulation stage, it may take 65 days, and most of them undergo atresia, and very few follicles reach the stage of ovulation. So, to complete this, this is the ovulation physiology. As you all know, so the estrogen, once a follicle is recruited and it is developing, the estrogen level will increase. And once the estrogen level crosses 200 to 300 picograms and for about 50 hours, then the LH surge happens. So the LH surge, what does this LH do? It will increase the progesterone hormone in the follicle. The progesterone will lead to follicular hyperemia and the prostaglandin secretion. There is a activation of proteolytic enzyme and which causes the weakening of follicle wall. The plasma will transuded into the follicle, which causes a follicle swelling, and finally the follicle rupture happens with the evagination of ovum. That is how the ovulation happens. So thank you.
I think.
So anybody has any questions?
Madam, like the follicular development as such, it takes 65 days, we are telling right now in the last picture you have shown. So if you see a patient and you encounter that, like males, if we have any sperm semen analysis abnormal, like we wait for at least 3 months would be an ideal thing. Like that, do we follow, I mean, like in your daily practice, do you follow it for females for a proper egg to see that egg is developing and all? Or you see that if the egg is not developing well, would you like to just down regulate with any OC pills and then rebound ovulation and all, you call her later after 1 or 2 months?
It is depending on the patient. So for every patient, we don't need down regulation. Okay. So for every patient, when they first visit a doctor, fertility specialist, so the main thing is the ovarian reserve, what we have to see before giving the time. Ovarian reserve, her age, so what are her previous treatments? What is the previous treatment? Has she undergone any ovulation inductions? So how was the follicular development in those cycles? So depending on that, we need to decide. So whenever a patient comes to you, if she's young, ovarian reserve is good, and she has regular cycles, so which means that most probably she is ovulating, regular cycles, young, sperm factor is okay, tubal patency is okay. Then if she's normally ovulating and if she's not able to conceive, then we give stimulation, that is for super ovulation. She's already ovulating, so it is not called ovulation induction. We give the medicines so that she might develop more than one follicle, more than one follicle. If she ovulates, then she'll have a good chances of pregnancy. So it is not that we will give 2 months time and then ask her to come back, or down regulation is not required for every patient. So depending on her problem, what is the condition, her ovarian reserve is very less, then we will advise her. Then what is the, if no other factor is there, but still she has very less time, so we will advise her what to do next. Then, if she already has some other problems, if the male factor is also there, age elderly, then we will advise the uh, higher methods for pregnancy.
Are you in your regular practices following this random start ovulation stimulations, man? Like they might come to you at any phase of the cycle. So
Correct.
you're not waiting, like depending on if she needs higher treatment and all.
Yeah.
So yeah, you are following that, madam, like this
Yeah,
theory.
we do random start stimulations also. Uh, and we are also getting good results with the luteal phase stimulations, especially.
Mm.
So, as we see, random start stimulation, they were started in patients where the patient is undergoing some chemotherapy for cancer patients. So, for them, as they don't have time, so they have to start the stimulation as soon as possible. So, once the egg pickup is done, they can undergo the chemotherapy. So, as studies were done in this population, where the random starts have been done, the oocyte quality, the blastocysts, everything was okay. Euploid rates, everything was comparable to the normal conventional stimulations what we do. So we have also started doing the random starts in the normal population, and we see, especially for the poor reserve patients, the elderly reserve, poor reserve patients, we get good number of oocytes. And in the same patients, we do sometimes the conventional day 2 stimulations and the luteal phase also. In the
Yeah.
same patients where the reserve is very less, and when we compare, the luteal phase has got a better in some patients, better blastocyst conversion in few patients. So when the patient has very less time, for especially the international patients, so or the husband stays for 1 month or 2 months, they want to finish the embryo transfer as soon as possible, then we need not wait. For all patients, we need not do random start, but for who need it, for patients who need it, we can straight away start.
Yes, ma'am. Thank you.